Fine now?
Yeah, now it's look good.
Okay.
Yeah, fine. Thank you.
Okay, thank you. Yeah, sorry about the glitches here, but I guess we can get started. Just yes, to dive in deep into that, I'm talking about exposome effects on the microbiome. So just want to give a public service message here. I just want to recall service done by the first responders, healthcare workers, and community workers from around the world at this time. I just was in a quarantine, sort of in a quarantine period here as I arrived in Chennai very recently. But I'm recalling the 100 days I spent in New York City like, in the recent past, where we were really badly hit by the pandemic. And I just want to put out a message saying that please make sure you wear your masks, and that way you can cut transmission and you can even reduce the rate of spread, especially out of concern for people who are out there and their community workers taking care of us. And just a shout out to community health workers and everybody around the world who are really helping us at this juncture. Just want to start off with acknowledging the people who matter the most, because typically we tend to miss out on those people who have impacted your careers and your lives. And I want to start off by acknowledging Professor Rob Knight, who's my mentor from my postdoc days, who actually introduced me to the field of the microbiome and gave me an opportunity as a postdoc after I did my PhD here in India, as well as several other mentors, starting with Chris Mason, who's the founder of Metasub, the lead and the brainchild behind the whole consortium effort from Weill Cornell Medical School. Mark Hernandez, a mentor, postdoc mentor at University of Colorado Boulder, and specific training on bioaerosols and working in the built environment. And Professor Peter Groffman, Zhongqi Cheng, Dr. Theodore Muth from the Graduate Center, City University of New York, my PhD advisor, G. N. Hariharan, who's Executive Director of MS Swaminathan Foundation, Professor MS Swaminathan himself, who was a doctoral committee member of mine. I'm indebted and very influenced by some of the value systems that he held and always instilled in research scholars for all of us. And several other collaborators globally from several of these projects that I've been involved with, starting the Earth Microbiome, the Metasub Global Consortium, and also multiple other collaborations. I just want to start off, especially since I'm talking to students, on some of these quotes that have moved me in my lifetime as well as in career. And just to think about it as something, as a message to learn by standing on the shoulder of these giants. And to start off, I want to quote Richard Feynman, the Nobel Prize winner in physics for quantum physics in 1965. He states a very profound, yet simple but very profound statement about the key to science. It doesn't make any difference how beautiful your guess is. It doesn't matter how smart you are, who made the guess, or what his name is. If it disagrees with an experiment, it's wrong. That's all there is to it. This statement is immensely profound in a way that it actually communicates how you should be practicing science and how you should be driven towards addressing your questions about science. And it's sort of very motivating in that front. And at the same time, there's also this caveat that you can derive from the next quote that's from the most famous Albert Einstein, which is about insanity is doing the same thing over and over again and expecting different results. So some of these quotes, I think, will make context to what we are going through in the course of the pandemic that's happening around us. But at the same time, it also gives you a primer of what you need to understand or how you need to be practicing or actually planning for research endeavors or even working together as a scientific community. And then lastly, I want to quote Tom Cech, the Nobel Prize winner for the discovery of ribosome in 1989, who states that we are stronger together when we are a team. And scientific research is a team effort, and it's not a single unison process which goes from individual to individual. And none of these projects that I'm going to talk to you guys about would have happened but for team effort. It is not a single person's endeavor. And we work and benefit as a team when we each of us contribute in a very efficient manner where we understand how to rationally approach the problem you're trying to solve and go driven by data, driven by experimental evidence, and not based on prejudices or notions or pedigrees that are swaying the thought process of science. So we need to be self-aware and be practicing that. To that note, I want to reflect on this infographic that came out of a paper in 2015 that talked about human perturbation of our planet Earth. And one of the major challenges that we are facing as a global community are in these areas of climate change, biosphere integrity, and biochemical nutrient flows. So if you look at this figure, highlighted in red are these critical zones of nutrient flow, starting with nitrogen and phosphorus, and looking at land system change that is interspersed with genetic diversity, or the risk for reduction in genetic diversity, which means you have species reductions that are happening in separate pockets or habitats around the world that is posing challenges. It's posing challenges for other species where they're not able to have access to otherwise accessible areas. This actually holds profound significance in the current context. And if you think about it, there have been signs that we have seen in the environment around us, starting with atmospheric abnormalities that have happened due to habitat changes. This is a picture of the Dust Bowl in the 1930s in the Midwestern United States. And what happened was, due to mass destruction of forest cover, of land, or domestication of the land for practicing agriculture or for doing other endeavors, the soil crust cover was disturbed. Thereby, it caused this wide swaths of dust storms that was tremendously impacting the ecosystems around the area. So this is an image of people from Kansas, the Midwestern US, or Southwestern US. And the way they are dressed is something that's reminiscent of what we are going through right now. But this is for an entirely different context. Although the origin of what has happened is actually very common. It's a very common problem that has happened. It is destruction to habitats, destruction to environments and ecosystems, which actually creates these abnormal exposures, or the events of exposures. The reason I bring that up is to put it in context. This is something that came out yesterday about rare earth mining, which is a habitat destruction mechanism that's happening, has devastated the environment in China, in mainland China. And the reason this has happened is because of demand in developed nations for these rare earth metals and materials that would otherwise be the ingredients for you guys having a fancy phone, right? An iPhone, for instance, has a requirement for very specific semiconductor devices that would otherwise not be found unless you find a very specific rare earth from a mine in a different continent altogether. So there is this rat race from each of these countries, developing nations, that are competing for contracts to mass produce such commodities, thereby destructing these land formations. And that poses challenges to the species that are co-occurring in those habitats. Lo and behold, we have one such problem with bats. And the problem with bats has stemmed to the fact that their habitats have been challenged. They have had human incursions into their habitats. Thereby, the humans are being exposed to those excreta potentially coming out of these bats or secretions from these bats. So one such event that had happened in 2012 in a mine shaft in a place called Mojiang, which is in southwestern China, came up because there were 6 men who went into those caves for reclaiming or kind of cleaning purposes, and they were affected with a pneumonia-like illness. And 3 of those men died because of that pneumonia-like illness. And the 3 others were all in ICU. They struggled through a respiratory distress syndrome. They later on found that the virus that had entered these men was called RABT-CoV 4991, which they now state is the closest genomic relative to SARS-CoV-2 and has 96.2% similarity. This is the source here I quote is the Daily Mail in UK. And at the same time, I want to quote another agency that is at the forefront of fighting pandemics. The EcoHealth Alliance is a nonprofit based out of New York City. They are in Manhattan. They have been here for a couple of decades. They have been focused and been working globally on projects associated with surveillance, with prediction, and also trying to study the specific cause of spillover events for pathogens, for these viral outbreaks and outbreak events that happen. Peter Daszak leads that institute, and he's a very famous British animal disease expert, and he states that the original assumption that SARS-CoV-2 originated from Wuhan is flawed, and it probably emerged from a different spatial location. The facts are out there, but I'm not stating anything to initiate conspiracy theories here, but it's for you students and researchers to comprehend as to what the challenge really here is and how certain facts are being distracted or being diverted from our attention. So the challenge here is habitat destruction and what exposure events can happen due to habitat destruction. At the same time, we need to be cognizant that one thing that they have really understood right now is we still are incapable of dealing with the impact of such pandemics because of the lack of standards, global standards or consensus between research groups working in different continents. And that actually came out in a recent paper that came out from my colleague from the Metadata Consortium, Lynn Schmill, who works for the Genomic Standards Consortium, brought out this paper on scientific data about COVID-19, revealing the peril of ignoring metadata standards. So metadata standards is something I'm going to be focusing on also in the context of the Earth microbiome that I'm going to speak about. So we are faced with threats from zoonotic pathogens, and there is a tremendous viral onslaught. And if you look at this figure on the y-axis, you have the number of basically emerging viral diseases annually, and this is plotted over decades. So starting with the number of decades, starting off in 1945, we have had an increase in the number of these zoonotic pathogens and viral pathogens, starting with HIV that spiked in early 80s or late 70s, and we have had outbreaks for the Nipah virus, avian influenza, SARS in 2002, H1n1 influenza. Again, you had events such as MERS and Ebola that are happening in the intermediary period. And I had a chance also to work on the H1n1 domain a decade ago, actually, when I was with the contract research organization here in India. We're working for clients in the US. But it's actually driven by the population expansion. Close to 1.6 billion people were living on this planet in 1900s to now 11.5 billion people in 2100. And we are increasing the encroachment into wildlife habitat spaces. This is resulting in these spillover events of novel viral threats from wildlife that are spilling over into human beings. And this is a problem that we saw coming in plain sight. There is this paper in Lancet that EcoHealth Alliance actually published in 2012, where on the y-axis here, again, they are plotting the number of cases with exposure events from viral diseases and looking for, basically this was a meta-analysis, looking for spillover events from animal into humans, which is mediated by these vectors or mediated by proximity to domesticated animals that otherwise come in contact with human beings. So they had an inkling as to what is happening and what could happen in the case that you have this mass spike that would occur when you have the right conditions for the pathogen to become even more virulent and become a pandemic strain. So we're now understanding that SARS-CoV-2 that causes the COVID-19 infection doesn't only affect our lungs or the pulmonary system. There's a very neat review that just came out last week in Nature Medicine from Ankriti Gupta at Columbia University Medical School, where they talk about the implications that SARS-2 can do, basically CoV-2 can do in the neurological space, in the renal space, hepatic space, gastrointestinal space, endocrine space, or cardiac space, or thromboembolic space, or dermatological space. So there are multiple manifestations in different organ systems. And the reason I'm bringing this up is spatially there is a distribution, which means in every different part of the human body, this virus has created havoc when it gets exposed or when it gets internalized. It has got the capability to basically access any portal. I mean, if you just want to call it in a very sci-fi manner, and even the brain, which is actually considered to be the least permeable portal because of the blood-brain barrier. So now, and even the GI barrier, which is actually one of the most robust systems, which we consider the second brain, and that is shielded by the microbes that we have in our own bodies that are actually our primary defense mechanism that prime our immune systems. And there is no better time than to focus on those organisms that are helpful to face the onslaught of such pandemic strains or pandemic organisms of virulence that can attack us. And we are living right now in a very silent spring for immune disorders. If you look at this figure that came out in NEJM from Bach, we're looking at the decades from 1950 onwards. There is a drop in the rate of infectious diseases, starting with rheumatic fever, measles, or tuberculosis and hepatitis in mumps, although tuberculosis will not hold good for India or Asia, or countries that are not that advanced in tackling that. But for other countries. And in the case of immune disorders, there's a rate of increase if you look at these conditions like asthma, type 1 diabetes, Crohn's disease, and multiple sclerosis. And all those categories of diseases are attributable to immune dysfunction that has happened because of changes to the microbiome or the composition and the functionality of these microbes that live on us, inside of us, and actually interact with our own genes to help operationalize several critical systems. And the reason now we are able to even understand or know about these organisms is because of the explosion in technological advancements. And this is because of sequencing becoming more cheaper over time, and also the advancements in the types of sequences that have been developed. So we can think of these sequences as molecular microscopes. The way Anton von Leeuwenhoek was able to visualize the first set of microbes in the 1600s, now we are able to visualize much more than ever from the fraction of microbes that we otherwise thought we couldn't culture on a petri dish. We are talking about these non-cultivable or uncultivable microbes that are dark matter, that are considered microbial dark matter, that now we're able to resolve using sequencing technologies, so much so that we have handheld devices like the Oxford Nanopore or the MinION that we can use in outer space, for instance. They have conducted sequencing experiments in the International Space Station. And in fact, Dr. Ganesh and his mentor, Dr. Kasturi Venkateshwaran, who I want to acknowledge at this moment, have been like from the NASA JPL labs, have been collaborating with several of these fascinating projects, including with Chris Mason's group that's leading the effort on epigenetics and microbiome in the space. So let's reflect back on how we try to understand from sequencing information specific to the microbiome. We always put that in the context of evolution. And primarily, we want to look at the 3 domains of life. If you want to consider microbes, you want to talk about the 3 domains of life, primarily because those domains are dominated by bacteria, and they're dominated by archaea, as well as the eukaryotes, where we will also fall under that category. And in this case, I want to quote my mentor, Professor Norman Pace, who's actually, I would call him the godfather of environmental microbiology and metagenomics. Every species alive today has been perfected for its niche through millions of years of evolution. And evolution in this case is a radiative pattern, and it's not a ladder, which means that you can have multiple radial tangents or radial lines that you can draw the way organisms can differentiate from each other. This is basically driven by the phylogenetic information that you derive from a gene that's been conserved through time in these organisms. And most recently, because of advancements in sequencing, we are able to look at specific genes as markers to understand how conserved or how similar species are to each other, or how different they are with each other. So initially, a lot of the work that Norm Pace's lab had done was looking at these marker genes like 16S ribosomal RNA, which is a gene that is highly conserved and has several hypervariable regions that can be used to study the evolutionary conservation elements between bacteria and archaea. The same would hold good, but in the case of fungi, you have an 18S ribosomal RNA that you can use for studying these marker genes for establishing elements such as phylogeny or looking for inferences between species differences. But at the same time, this technology was not robust enough to provide information on something that you can go down to the strain level of the individual organism, and at the same time also looking at functionality of the organisms, which is why we have advancements that have been done from the genome sequencing processes that have yielded a technology called metagenome sequencing or shotgun metagenome sequencing, which is more robust in providing that enough amount of resolution. And using such genome technologies or genome sequencing abilities, we now have a very new tree of life, where this group from Jill Banfield's lab in University of California, Berkeley, came up with this new tree of life where they show that archaea are actually a very interesting group or domain of organisms that actually act as a transition between us and bacteria, or eukaryotes and bacteria. So the eukaryotes are interspersed in the archaean clade in this case. And they wouldn't have been able to derive this information but for these advancements. And we now have close to 92 bacterial phyla, 26 archaeal phyla, and 5 of those eukaryote supergroups that are branching within these superphyla of archaea. And you always have these uncultivable taxa that keep dominating the tree, like the candidate phyla radiation, where some of them are unresolved, and you still need to sequence even more or derive more assembled genomes that you can help inform and further refine this tree. I'm sure in another couple of years from now, we will find an even more resolved tree where you can have even more information on what is actually closely related to what. So from such approaches, what happened was they built the first map for the human microbiome. This paper came out in 2012, and at the time I was with Professor Rob Knight's group, and Rob's group was very actively involved in the NIH Human Microbiome Project. A lot of the analysis was being done in our lab at that time. And this figure actually came out from Curtis Satano's lab in Harvard with Nicholas Segata, where they were able to show 4 most abundant phyla in the human microbiome. And they actually came down to 4 different types: Actinobacteria, Bacteroides, Firmicutes, and Proteobacteria. But interestingly enough, if you look at these clades, or if you look at the distribution of these dominant phyla, they had very specific niches or very specific preferences for where they were dominant. For instance, if you look at something like the Proteobacteria or Bacteroides are more abundant in very specific habitats. Like you can find them more abundant in the gut. And at the same time, if you find groups like Propionibacterium acnes, that's something that you will find on the skin, and it causes conditions like acne. So there are very spatial preferences and functional preferences for the way these organisms are located in different body sites, starting with the mouth, with the gut, or with your skin, or even your reproductive organs. So that's the way it's very distinct, and it has very spatial preferences for that. So when you think about the onslaught that I just talked about, right? SARS-CoV-2 invading us and percolating in different organ systems or different areas in our body, it basically means that the virus has established a communication pattern with these invisible organisms who have a preferential way of positioning themselves in those areas, right? So we are now more aware of the fact that we are in no way uniquely human by ourselves. We are encompassed by the microbial cells or the microbial organisms that live inside of us. And if you think about the number of cells, of microbial cells, there are 100 trillion of these cells, and which is comparative to just the 30 trillion of human cells that we have in our bodies. And at the same time, if you look at the number of microbial genes, they are overpowering us by several orders of magnitude. And the human microbiota can be categorized into these elements. Basically, it's bacterial microbiota, or the bacteriome, or the fungal microbiota, or the microbiome. And you also have microbiota, which is human microbiota, which is constituted of the viral microbiota, that is virome, as well as parasites, such as the helminthine parasites that are also there as part of the microbiota in our guts. This is more specific to countries that are underdeveloped or in developing nations, because in developed nations, they are not as much as exposed to the parasites than what we are in growing up in countries like India or Southeast Asia, which actually, if you think about this fact, it becomes very interesting, because one of the initial drugs that they said that had efficacy in reducing the rate or the intensity of infection for COVID was a drug called ivermectin. A group in Australia was able to show that. And ivermectin is a drug that's given as an antiparasitic, and in an antiparasitic regimen. So it's very interesting to see that people whose system have been exposed to these parasites could potentially have a protection against drastic reaction to the onslaught of viral triggers or immune triggers. This is a hypothesis that still there's a clinical trial that's ongoing, but it's interesting to think about it in this context. And I want to focus a little bit more on the virome in this point, because we're talking about a virus, a viral pathogen. If you think about the virome, there's still so much that we don't know. There is still so much that is mystical about these viruses. And I know that in the seminar series earlier, your faculty have also had some interesting talks on these topics, and I've been following that as well. And if you think about the number of bacteria versus the number of viruses, it's different orders of magnitude. You look at 380 trillion viruses that are making up your virome. And only recently have we had the capability to even sequence and assemble virome sequences together. I want to acknowledge the efforts of Professor Forrest Rover from San Diego State University. Forrest Rover used to be a collaborator at Rob Knight Lab during his time at the University of Colorado Boulder. And his effort resulted in the first assembly called CRASPhage, which is cross-assembly sequencing approaches to assemble a bacteriophage. He worked a lot on coral reefs, on marine environments, and that was where a lot of the virome efforts were initially pioneered. And now we have the capability to use DNA sequencing and enable prediction or enable contig analysis for DNA viruses using the JGI or Joint Genome Institute's IMG virome database. So right now, this is a paper that came out in scientific reports where they were showing close to 750,000 DNA sequences that you can attribute as to having total viral information. This is specific to DNA. I'm talking about DNA viruses in this case. Because for RNA, you'll have to do metatranscriptomic approaches where you're going to sequence from the RNA and then go from there. But in this case, this database is rich for having unique viral sequences to the tune of 400,000 right now. So they're still improving this, but it's actually a huge resource, which is open source and can be used for research. So after the viral part, I want to dive further into the part of the microbes or the bacteria here, where we now understand that microbes can be categorized in our gut primarily by our exposure to the diets. The diet that we consume is going to influence the type of microbes that predominate in our guts and thereby influencing our health. So the primary categories here are Bacteroides or Prevotella or Ruminococcus. Basically, you can attribute these diets influencing groups specific to people who consume excess amount of vegetables, carbohydrates, and also have diets similar to hunter-gatherers. And the people who are individuals who are consuming more of westernized diets and consume more of meat have predominantly Bacteroides groups. And also you have people who are consuming, or groups that are consuming fiber-rich natural diets or traditional diets that have been following from age-old times. They are dominantly Ruminococcus. And very recently, there was this paper that came out in PNAS where they classified these groups into Team Prevotella or Team Bacteroides. So we as individuals coming from countries like South Asia or primarily in India, from the initial data that's come out on the microbes, especially the bacteriome data, we are very dominant with Prevotella, the group Prevotella, versus the Western diets and the diets consumed by North American counterparts, who are very predominant in Bacteroides. At the same time, one thing we need to be aware of is there is this concept of the environmental feeder effect or the environmental metagenome effect contributing to the host genome and the microbiome within the host. So they call this the Holobion Theory or the hologenome conundrum. I call it the conundrum because this theory is still debatable. There is a lot of if and then, or if not, why not kind of scenario there. Because they talk about the microbes that are constitutive in our own gut interacting with the environment and taking in feeder elements from the environment, from the microbes of the environment that are exposed to, thereby influencing how the symbiotic attraction or the symbiotic association for nutrient uptake or even for interaction will occur. So interestingly enough, the environmental microbes that we are exposed to would not form part of that, which means they are not part of the core microbiome or the microbiome that's succinctly maintained by from generation after generation, which means if you are coming from a specific lineage in South Asia, you are primed to have a certain category of core microbes, even if you move from South Asia into North America or South Australia. So you're going to have your own, you can call it a microbe lineage that you continue to maintain. And you derive this lineage from the time you're born, from your mother. And that's something that's so profound. So when you move from country to country, you can also transform your gut microbiome. My colleague from Rod's lab, Dan Knight's lab, basically published this work in Cell 2018, where they showed that there was a group of Thai immigrant people who moved from Thailand, southern Thailand, into the United States. And they saw that when they were maintaining their diets, which was traditional diets that they used to have in Thailand, they didn't see shifts in their microbiome. However, when they moved to the US, they started consuming more processed foods or several other additives from what they get in the supermarkets. Their microbiome patterns were changing, which leads us to assume that the more and more your microbes are influenced by these processed foods and meats that you consume or cultivate, the more it decreases your microbial diversity, thereby making you vulnerable to potential changes in the environment that will otherwise challenge your immune system, which means the priming effect or the efficacy of priming is lost. And this is a study that did an analysis on how you can map the human bacteriome, in this case, based on urbanization metric. On an urbanization metric, which means the per capita income in individual countries, they were trying to categorize between the dominant types of bacteria that you will find in the gut or in the skin or in the urinogenital tract or the oral cavity. I'm just going to take one example from India here. If you look at the gut samples from India, Prevotella is dominating samples from India based on the urbanization index. At the same time, if you look at the gut samples that come out of, say, a country like Philippines, or let me just go to North America, you're going to have more of firmicutes in this case, right? And firmicutes are a group that is also attributable to obesity or trends that associated to weight increase. So you have changes between country to country based on, again, not only the amount of economic income they generate, but also it is influenced by the dietary practices that are also culturally influenced from these nations. And if you look at the number of undernourished people in the world, this is a fact that came out, this is an infographic that came out from FAO very recently. The number is actually on the rise, which is weird because we boast about increasing agricultural capabilities at the same time. And this number is going up drastically. It's now at 821 million number of undernourished people. And there's so much prevalence of undernourishment, thereby leaving us with the challenge of understanding what we are exactly consuming, which means we are consuming processed foods, we are consuming foods that are non-optimal, we are consuming ingredients that are loaded with pesticides, and thereby causing these exposures to those pesticides is making us challenged, or our microbiomes challenged. And fruits and vegetables are now at just 11%, whereas we need to be having 49% of fruits and vegetables on our plate based on Harvard's healthy eating plate model. And excessive amount of sugar is being consumed, thereby also challenging our body and other conditions physiologically. And as we know, microbes are ubiquitous and everywhere in the food system, starting with agriculture, soil. There are several interactome effects, starting with the exposures that you have in the environment, to the foods that you consume, the water bodies we are exposed to. And these are all having trickle-down effects that we would actually constitute as what they can call the One Health phenomenon. And one of the overarching goals was to understand, if you put ourselves as a global community and you look at environmental spaces, what types of microbes are there in the environments around us in these different continents. So that fact actually motivated Rob, my mentor, to launch this project called the Earth Microbiome Project in 2010, which was actually a very outlandish idea. It was very challenging. So he, along with Jack Gilbert, Janet Janssen, Jack is also at University of California, San Diego right now, and Janet is at the Pacific Northwest Labs in Seattle. They constituted this project where they thought, okay, let us collect environmental samples from each and every continent and do that in a standardized way, which means literally every lab and every researcher coming on board to do this project has to follow the same protocol to the T, and nothing can be changed between these protocols. So when in that case, we can replicate or we can find correlations between each and every habitat where we collect the samples from. So this project started in 2011. This was my first postdoc project as part of research with lab, with Rob and University of Colorado Boulder at that time, and we generated 27,751 samples. This was based on marker gene sequencing, at that time 16S sequencing, because sequencing costs were really high. And we were able to collect samples from 7 continents that included 43 countries and close to 500 plus scientists. And we were also able to collect basic metadata from each of these sites samples were generated from. And that resulted in a giant reference database of bacteria that reside on planet Earth. And this is the largest cohort, which is open source, and it got published in 2017 in Nature. And this paper is free, open access, and the data set is also open access, which actually can give you information on the host associated environments as well as free living environments, such as soils or estuarine regions or glaciers or deserts, for instance, or coral reefs in the marine areas. So this was the EMP's goal, and with the data, one thing that we found that was staggering was there was predominant amount of data we derived from soil samples, and close to 6,000 of those samples or 5,000 of those samples came from soils, as well as we also had, this is from the free living space, and also from water samples, close to 5,000 of those from water. And if you spatially try to understand how they interrelate with each other, we were also able to have metadata categories that gave basic elements such as pH. It also gave information such as site where the samples were collected from the host, in this case host plant, or whether it was the host root, plant root, or whether it was the soil, and such elements associated with that. So this is basically a figure that, in this case, you have millions of dots, or rather thousands of dots on this figure. It's represented as a principal component axis plot. So what these dots exactly represent are the sequences for 16S ribosomal RNA that are identified with each sample. And the plot distance basically measures the distance between sample A and B, in this case, or sample N, using the phylogenetic similarity. So for that, the NITE lab had developed a technique called UniFrac, which is a unique fraction value index that essentially measures the phylogenetic relatedness between species in a particular habitat or between habitats. And the phylogeny is determined by literally by the phylogenetic tree and how these sequences are common or different from each other, or whether they overlap with each other. So you can see that soil in red on the lower panel of the figure is actually very diverse and very different from something like an animal secretion in light brown or animal surface, which means soil is a free living ecosystem entity, and the animal surface or animal secretion is a host associated entity. So these are governed by several other variables that will influence the composition of microbes that you find in those ecosystems. Thereby, we were able to see that mass change. And at the same time, in soil, we also found plant rhizosphere, which is the region that's close to the plant root, but in proximity to soil. That actually turns out to be one of the most diverse habitats along with soil, which is actually a staggering fact that we could unravel. Because of which we know that that is the site which primes the way your crop is produced or the way you grow your food, which is actually very important. And that is the way you sustain the health of the food, as well as sustain the value of life in this case, because you need to have the optimal set of microbes that are maintained in the soils and held back in those areas of your plant. And the abundance of such microbes between the host and the free living, again, if you look at the crosswise comparison, in this case, abundance in the y-axis for 16S ribosomal RNA sequences, plant rhizosphere is actually peaking up along with soil. In fact, you look at more amount of richness when you look at the plant rhizosphere surfaces. So that project from the Earth microbiome on the environment became the backbone for establishing what became the largest citizen science microbiome project, human microbiome project, called the American Gut. So this was built on the Earth microbiome project based on the protocols we had standardized for processing samples using these amplicon approaches or using the marker gene approaches. And now it's grown into a global initiative. I want to acknowledge the efforts of Daniel McDonald, who's my colleague at UC Boulder, and he's the director of the American Gut Project at UC San Diego, along with Rob. And they actually have ongoing initiatives such as the British Gut Project, the Microseta Global Initiative, where they are trying to become more inclusive. They would welcome participation from different countries and different researchers. So this is one such project that's ongoing. You can go to the website at UCSD and also look for more information on the site. And I also want to highlight, or if you students are interested more on this topic, to go and check out Rob's TED Talk, where he explores the capacity or the potential of the microbes and look at how they impact our lives and how they impact the environment around us. So just, he's got a book as well, which is called Dirt is Good, which is actually a very good read on this topic that you would like to refer to. So from the data that they generated from the American Gut Project's first phase and the previous data that they collected from the Earth microbiome, they were able to ask the question, how are we humans connected to our environments, microbially speaking, right? Whether there are any linkages. And they were able to answer this question in a phylogenetic framework, or looking at the phylogenetic relatedness between samples. So in this case, on the plot on the top left corner is again a similar inference of between sample information, starting from samples that they collected in the Earth Microbiome Project, projected with the American Gut Project, which is host associated. And EMP is primarily free living, but also has certain hosts which not at all human. Everything but human on the other side in green, but in brown you only have human. So you have this intermingling effect. You have this overlap effect between human host and the environment. And that they were also further able to unravel on these phylogenetic trees in the figure B on the bottom panel, where you were seeing clades like Firmicutes, or Fusobacteria, or even Bacteroides and Proteobacteria getting interspersed with the Human Microbiome Project data samples, with the American Gut Project data and the Environmental Project data samples. Interestingly, some of these phyla are influencing several processes in the human microbiome. And Fusobacteria, for instance, has now been implicated in conditions like colon cancer. This is actually getting unraveled slowly now. I'm not saying that the initial framework from AGP was able to discover a biomarker there, but these are basically signals that can help inform follow-up studies where it can be done in a more concerted way. And the take-home from that also was this other inference where they were able to understand between industrialized communities and hunter-gatherer communities or remote farmer communities how there is a variation in these bacteriomes, which means you have very specific niches or specific types of bacteria that predominate industrialized communities like black nose spidaceae, for instance. Or if you look at these communities like Rickenellacea, which basically are only found in the industrialized and not found in these remote farming or traditional dietary, people who practice traditional diets. And some of these taxa, such as Ruminobacter or Treponema, otherwise Treponema is something that you're exposed to only when you're exposed to parasites, or only when you're exposed to being in the proximity of livestock or wildlife. Whereas if you look at the modernized communities, they have no exposure to livestock or wildlife, which actually is not advantageous because the system is not primed well enough to make them robust to be able to cope with that, right? So that's actually one of those inferences there. And more than ever, we need to be cognizant and understand this concept of the One Health relationship, where we human hosts are influenced by the feeder effects from the environments, chemicals, the diets that we ingest, the environment around us, the animals we are exposed to, the pets that we have at our homes that also influence our health. And we are not a single entity. We are dependent on each of these modules in the environment around us. And the way we respond to those effects or those signals is going to influence our overall health and overall ability to cope with pathogens, cope with these challenges that we are faced with right now. And now more than ever, we need to be aware of how we best prime ourselves or position ourselves to cope with these challenges, thereby not interfering with the basic mechanisms or the basic ways we are empowered by these microbes. I talked about the primary microbial lineage coming from our mothers. From the time we are born, we are exposed to several other variables that can influence how we develop over our lifetime, starting with the process where we are hospitalized or where the way we are born, whether it's via C-section or vaginally. That also influences how well we cope to the environmental exposure. So it's also been shown that children born via C-section are more prone to respiratory disorders or asthma, conditions like asthma. They're also talking about cognitive conditions like autism, for instance. And also when they are not exposed to the right diet types, for instance, when the infants are breastfed for a longer time, they have greater protection because they derive very unique set of immunoglobulins. They also have probiotic bacteria that potentially are helping them cope with these environmental pressures. And right now you have a process of formula milk where you're also having supplementation that is being provided. At the same time, there is also this constant influx of antibiotics or these medications that have been meted out to us from a very young age. And sometimes there is an overdosing of this that's been happening. And there are also this fact that 20.5% of babies are suffering from low birth weight. And I want to touch upon this topic of antibiotics and antimicrobial resistance more specifically. And in this case, I want to acknowledge the talk given by Dr. Sai Subramaniam, which is actually very, very helpful. And in fact, you have primed the students for me beforehand, and thank you for covering several of those topics there. But this will actually be helpful for me to kind of push that forward and try to help them understand the role of AMR and the challenges that AMR poses in this current context. So I want to highlight one such study that came up very recently, last month, from a collaborator of the Metagenome Consortium, Dr. Niranjan Nagarajan at A-Star Singapore, Genome Institute. And they published this work in Nature Medicine on the cartography of opportunistic pathogens. And Dr. Sai talked about the escape pathogens, which is actually one of the most studied group in the hospital-acquired infection space and also in the multi-drug resistant category. However, we also have microbes or pathogens that are not in the escape category that can become antibiotic resistant, that you can only track down using next-gen sequencing methods, which is exactly what Niranjan's group has pioneered. And they were able to do a survey of hospital sites, and they were able to also show how these infections start in a hospital site. And this is work done over a couple of years where they collected multiple samples by using three approaches. Basically, it was a direct shotgun metagenomic approach, Illumina sequencing enabled. They also did a culture-enriched approach for long-read metagenomics by basically collecting samples from these sites and then plating them and then resequencing them. And they also had a backup set that they were using for DNA extraction in this case. What was novel about this is they were able to collect from different time points. They started off in 2017 November, went all the way to 2019 May. And they collected from 179, several other sites. Basically, cumulatively, they were able to do close to 500 sites in this case. And they were able to recover unique number of genomes, in this case, closed plasmid seq genomes close to 5,900 and several other genomes. One second. Yeah, 2,347 of these genomes and 1,600 of phage sequence genomes. So what they did was they also were able to establish the relative abundance of some of these escape pathogens. In this case, they were categorizing them based on the abundance of, say, Klebsiella pneumonia in isolation rooms or in multi-drug resistant organism wards, specific wards, or even in the standard wards. They were able to also categorize it on that basis. So if you look at something like Pseudomonas aeruginosa, they are more predominant in these isolation rooms. And something like Staphylococcus aureus, where you have the very, very lethal multi-drug resistant types, or in the MDRO wards. That's something that they were able to also understand. And one interesting thing they were able to show was distinct ecological niches within hospital environments, because the hospital room itself is like pretty much like a mini micro ecosystem, because from the place where you have your sink tap to the place where you have your pulse oximeter, there is going to be a variation in the type of species that you will find, and also a differential abundance or a difference in the abundance of specific type of microbe from one site to another. That was actually one of the first times they were able to show that with that level of precision. And in this panel on B, you can look at the differential abundance between these species such as Acinetobacter or Corynebacterium or Staphylococcus versus ones that are pretty much environment associated, or in this case, Methylobacterium or Burkholderia that you will not find in a closed ecosystem. It usually is associated with soils or in environmental habitats. So that leads me to another point where that study is actually, there's further more information available. Please refer to that paper for even more additional results. But I wanted to focus on this topic, which also Dr. Sai mentioned, on the metallobeta-lactamase, because these are these enzymes that are impacted and can confer resistance to antibiotics and the way the antibiotics can act, right? So there's this case of the New Delhi metallobeta-lactamase 1, and most recently, this was discovered a decade ago in India in 2011. And most recently, they found this from the Arctic, from soils close to northern Norway. And a group led by Dr. David Graham was able to show that, from UK, he was able to show that they found that this gene BL8-NDM1 which is actually the one that will make the protein for NDM, the metallo-beta-lactamase, was found in that area. And it actually stumped everybody as to how did it get that far, right? How did it get that far as the Arctic Circle or as northern Norway? Because originally they thought they found it in New Delhi. So this is a moot point because they later realized that you have birds and wildlife or that could be exposed to these plasmids that could have transferred it via an aerial route, which again leads us to ask the question, was New Delhi originally the origin for NDM1? This is something, again, I'm putting out for debate. It's up to you guys to also talk about it. Okay, so I'm reflecting on another study that was follow-up, or basically the study where they collected samples from sewage and drinking water after the 2010 Commonwealth Games in New Delhi. And these are in green. Some of these sites, both sewage and drinking water samples were collected. These sites included sites close to the Asian Games village here on the bottom side of the figure, as well as the one I've encircled in red, which is actually the Commonwealth Games site or the Games village site in 2010. And they extracted the sample in 2011. Please pay careful attention to the timelines here. 2011, and they looked for this NDM1 positive bacteria. They were able to identify the plasmid to that. And in 2013, 24 months later, they're able to find this as far away as Svalbard in Norway. So they collected these samples. They went back and processed these samples. So when you're talking about Commonwealth Games, it is a context for mass exchange of people coming into one particular city. Several Commonwealth nations, athletes came into the city. Several tourists came into the city at that given point in time. So it is not originally driven by something that happened from within New Delhi. It is also the fact that people need to acknowledge or be cognizant of the fact that there were feeder effects from several continents as well. This is something that has not been discussed further, but it actually gives us a scope for understanding such problems. And the worldwide distribution of NDM1 positive enterobacteria strains largely was relegated to countries like India, Pakistan, Yemen, Serbia, and Egypt, and pockets of these nations. But they thought that this was not prevalent in other areas on the globe. However, when they went and looked for the BLA NDM1 carrying plasmids in Enterobacteriaceae, they were able to find replicon types of that gene in several other continents. And if you look at those continents, this is a study that came out last year, reported that last year. If you look at these continents, you will curiously find several nations that form part of the Commonwealth on that map. New Zealand, Australia, South Africa, Zimbabwe, Canada. I can basically keep listing now on UK, this country, Scotland, and these countries that would have otherwise had people who were in New Delhi at the time of the Commonwealth Games. So I'm not saying, hey, this came in because of people coming in from other countries, but I'm saying that there needs to be mechanisms put in place to understand how these exposures occur and how these get disseminated from point A to B. So how do you do that? In that sense, there needs to be a platform where you're able to track basically marker phenomena such as antimicrobial resistance. And that was basically a motivator for the Metasub project. The genetics and epigenetics of antimicrobial resistance was a Gates Foundation effort that stemmed from an initial crowdsourced effort that Chris's lab put together from Weill Cornell. It was called PathoMap, where they wanted to study the distribution of antimicrobial resistance or the distribution of microbes or bacteria in the subways in New York City. And from there, they were able to have enough resolution where they wanted to expand this globally, because New York City is a melting pot for the world. Literally, there are people from several continents all around the world who come to that city, which is actually evident in the way COVID-19 million travelers flew in just from Europe in the month of January and February, when the virus was transmitting and being rampant at its peak. And we were basically hit down with mass spread of infection and mass spread of mortality actually at that time. So having the backbone of establishing vulnerability of populations two existing challenges like AMR will in fact help inform prediction or help inform authorities of surveillance measures they need to take, and also identify these areas that are challenged by the prevalence of already debilitating effects. Because if you have a population that's susceptible or succumbing or exposed to AMR, they are even more vulnerable in their ability to cope with COVID-19 or something like an onslaught of a virulent pathogen. And Metasub is a framework that can help complement such surveillance approaches. And major goals being geospatial, metagenomic, and forensic map generation, which looks at spatial dynamics, a very specific way, similar to the Earth microbiome, but in this case, this is focused on cities. This is focused on urban environments where people are all congregated, right? And understanding antimicrobial resistant tracing from these habitats. Thereby, if you do this, you're able to generate even more resolved microbiome information, because this is completely shotgun metagenome focused, and you have enough resolution to identify marker elements and also identify novel biosynthetic gene clusters that can facilitate development of new drugs from these data that you generate. And this is the snapshot of the first city-scale metagenome profile from New York City, from Chris's lab, led by Ibrahim Afshineku, who's actually the executive director of Metasub initially. A lot of the efforts from Ibrahim and Sophia on this study, in fact, helped build the backbone for several of what is happening at Metasub right now. I want to acknowledge their efforts here. And primarily, the framework is this. You do shotgun metagenome pipeline approaches from surface samples that you collect from these subways or metros. In this case, you do straightforward mapping back of reference genomes from in this case, it's a paired-end sequencing approach where you do like close to 5 to 6 million reads per sample, and you quantify the abundance of microbes that are present in each sample, and you identify the taxa or the type of these microbes from databases, database approaches. And then you also look at the functional repertoire or functional predictive ability from these samples. Thereby, in a sort of a nutshell process, this is what will happen. You're extracting DNA from these samples, and you're using these standard libraries, in this case, QIAGEN libraries. You do the alignment and confirm it with the database resolution. And also, you're able to have location-specific tags, which means at a given point in time, I'll be able to predict what type of microbe is present in which station in New York City. Right? And in this case, from the result that they found, they were primarily able to only identify bacteria, close to 46.96%. 92% was bacteria, but 48% of those was unknown organisms. So this one came out in 2014, on that time frame, right, when the databases were still not so robust. And this is just a visual representation of Pseudomonas density alone in New York City. If you look at Manhattan, the place in fluorescent yellow, the central part where close to 10 million people come in and out during the day for employment and get out. You can see that amount of intensity in this particular strain that you can see. And this is the kind of visualization that we are trying to provide. Also, in the case of the Human Microbiome Project, they were trying to compare how does this subway compare against the Human Microbiome database. And they found that a lot of the surface information that we find is similar to what you find on the skin microbiome. And coupled by these taxa like Staphylococcus epidermidis or aureus and acnes that you would otherwise find in the skin. And you don't find so much of the GI tract associated microbes on the surfaces of the subways. So this is a visualization of these laboratories that have participated in this project from the initial times. This is the plot from 2016 where we use one particular day of the year, pretty much it's June 21st, which coincides with the Ocean Sampling Day. We call this the Global City Sampling Day, or GCSD. And in a 24-hour framework, starting with New Zealand, Auckland in New Zealand, all the way to the west in Alaska, in a 24-hour period, swabs are collected from transit systems around the world in real time, and they are geosynchronized. And basically, they are fed into a common database. And surface samples are collected and aggregated in the same standards and standard methodologies and sequenced at central labs where we had coordinated efforts done by the Metasub lead coordinators. So this is something that has not occurred before in a very synchronous manner. And what has happened in the last 3 to 4 years is several of these data sets have been aggregated, and we took a subset of 4,700 samples from 60 cities around the world. And this is actually worked out in the form of an initial publication that we actually have in review. And there's a preprint available, but in distribution-wise, we have sites from North America, East Asia, contributing close to 1,000 samples each, and Europe as well. And in this case, we didn't have samples from India because at that time we launched it in India, but we still had roadblocks from the authorities on sequencing these samples. So it's just something that I'm trying to resolve along with my collaborators, who I'm just going to highlight in the slides to come. And from the major takeaway from that initial 60 site sample set was we were able to show that we were able to visualize specific types of bacteria that are commensal or that actually form biofilm from these sites. And each of these sites are different from each other. And you could see that some of these samples on the right that are there in East Asia are very different from samples that you find in North America, or there is sort of a transitionary shift in these samples. And these samples from Europe form this intermediate zone where the North American and European samples of concordance, so sort of a co-occurrence there that you see. This is again a Jacquard visualization, which gives you the presence or absence as well as the species overlap. This does not give you the phylogenetic relatedness, like what I previously showed, which is unifrac driven. In this case, this is Jacquard derived. So also from the same data set, we were able to project and show what is the microbiome diversity for these 60 cities, as well as the functional and the antimicrobial resistance profiles for these cities. On the top panel, you look at the prevalence of proteobacteria in purple and the prevalence of actinobacteria in red. That's constitutive of some of the firmicutes. Sorry, not firmicutes, sorry, the actinobacteria that you see, and the firmicutes in green that you find variable between regions like Middle East, or you find a variation between regions like East Asia, right? So you have more of those in Middle East versus East Asia. At the same time, if you compare the same Middle East versus East Asia, in the case of this is the metabolic functionality. In the case of, I'm just going back to the AMR class. That was one particular market thing that we saw. We saw the more of the MLS class against the North American, where you have a variation between these multi-drug resistant bacteria that you see. Sub-Saharan Africa was the most prominent with multi-drug resistant samples that you found from the cities in Africa, which means that it sort of gives us a moot point to understand the prevalence or the usage of over-the-counter medication that they have access to. When you include India into this data set is going to be very, very interesting. And because we have easy access to antibiotics and we have indiscriminate use of antibiotics as well. So that's going to actually inform this and resolve this in an even very more interesting manner. And across these city samples, we found 1,300 novel bacteria. 1,300 novel bacteria just from subways around the world. And if you go to those hyperlinks, you can actually go to each site, each city, and visualize what type of microbe is there or bacteria is there up to the phylum level. And that's something that you can check out. In this case, it's plotted within the novel versus known category, and also based on the spatial distribution of the sites. So in the left panel, you also have different classes of AMR and the correlation between each city, starting with Bogota and Colombia all the way to Sweden, Stockholm, and in New York City is included in that. So the mound on that graph basically reveals the prevalence of AMR classes, or the increase in AMR class aggregated through that samples coming out of that city. This is something as a trend line that you could see in Santiago, Chile, for instance. The mound is actually distributed all through, which means there's a continuous prevalence of, in this case, AMR classes that they are seeing. But this data could be even more refined into a specific class of these AMR attributes. For instance, these enzymes that are involved, like the NDM1 category I talked about. So we are also doing additional iterations of the data set, and we have several follow-up publications that are in the works that would be coming out pretty soon. And the other biggest takeaway from this data set was we were able to predict viral DNA sequences from cities that are sampled from Metasub. And we are predicting 10,000 novel viral DNA sequences from this data set. The way we were able to do this analysis was because of the JGI IMG VR database that I talked about in the beginning, where we were able to predict from DNA sequences resolved viral context that can be predictive of information specific to DNA viruses. And I want to place a disclaimer here. This will not, from DNA, you will not resolve RNA viruses. So we are not talking about COVID in this case at all. This is just DNA viruses. And that's the reason for COVID, we are having a parallel platform where we collect RNA samples from surfaces and we separately sequence that, looking for the metatranscriptomic contribution. So this is also a major takeaway from this effort. And this paper is available at the site. It's a preprint. It's actually in review. David Danko, I want to acknowledge, who is Chris's graduate student, is actually the lead, has done phenomenal work along with Daniela Besdan, who is the executive director of Metasub. True credit to go to the people who really led this from the forefront. So right now, diving into what's going on with India, the goal now is to establish a pan-India Metasub initiative. And I'm very pleased to say that Dr. Ganesh Babu Malimon is actually one of our collaborators for the Metasub initiative from Tanjavur at your institute, Shastra, and I'm really excited to have Tanjavur coming on board this effort. And we have sites in IIT New Delhi, IIT Delhi, and ISI Kolkata, in Hyderabad, Osmania University, in Chennai, IIT Madras, and even in Puducherry as well. So we're trying to see for a north-south gradient in this case, where we were going to have a two-pronged approach of swabbing for AMR from surfaces, as well as swabbing for COVID from these surfaces. It could be metros, it could be transits that are not metros. If you don't have a train system there, you can do it from buses or public spaces as well. And a quick snapshot on what work I've done at Hyderabad when I launched this 4 years ago, and also during the lockdown right now, my collaborator Someshwar Rao and his team, his lab students, have been able to collect samples. Hyderabad was very unique because we were able to collect from a brand new metro system even before the metro rail started. So we collected from a train that was not occupied by any passenger, which actually made it baseline data from Asia. The first site, the only site in India to have baseline data from a metro. That way it was unique. And we did sampling from surfaces, from trains, from these ticket vending kiosks. And these are pictures from the students braving the restriction during COVID lockdown, going out there with face shields and masks to collect samples. I really want to commend the efforts of Dr. Someshwar Rao's group and his students, his postdocs, and several undergraduates who volunteer as well, and Osmania University's cooperation in this regard. It's tremendous that they were able to carry out this effort. This is sampling done in 2016, where I was basically led into these clean metro trains where we were able to collect baseline data. We were using at the time Copan eSwabs, and we also did comparative data in these Mofussil trains that were highly crowded, and had some interesting conversations to passengers during that time because they were figuring out what we were doing, right? And this is sort of the stations that we collected from at that time. And it typically means you collect from these surfaces like ticket kiosks, ticket turnstiles, where you enter into the metro station. So these are the surfaces that we actually care about and want to look for, where people come in contact with them and what happens or what gets transferred. And this is the current status for 2020. Very recently, this happened globally last month. This is a photo of the team from New York City, where they were collecting samples from several swabbing surfaces. And then we have, like I said, several partners in the country. And we are looking forward to processing this set of samples. And hopefully we can get India's data out on that map and make it even more robust. And if you go to the site, this may be updated very recently, or they're still in the process of updating. You can see a snapshot of all these sites from where they've collected samples and how the visualization for what happened in 24 hours or what happened during this combined process of doing COVID sampling and AMR sampling and what's going on. And I want to take the time to acknowledge, again, Metasub teams very specifically, because this is a huge, enormous effort that we are dealing with. Brainchild of Chris, of course, Chris Mason, and Benjamin Young and Krista, who are, I'm actually a research affiliate with the Mason Lab, so we are all part of that giant team, and their kudos to their efforts and coordination across different sites. And the sponsors, as well as the government authorities in Hyderabad for L&T Metro, Government of Telangana, Narasimha Rao Nedunuri, who actually one of the key people who helped initiate the Metasub activities in Hyderabad, and all my collaborators from ISI, Kolkata, Malay Bhattacharya, Someshwar Rao from Osmania, Ishan Gupta, Delhi, Bhopal as well. Dr. Karthik Raman, Himanshu Sinha, and Indumati Nambi from IIT Madras, Ganesh Babu, as I mentioned, from Malimavan from Shastra, Dr. Damodaran from Puducherry, and the funding agencies such as National Science Foundation, National Institute of Health, Bill and Melinda Gates Foundation for the funding, Gordon Betty Moore Foundation, who actually had given me an early career award, Sloan Foundation, and these nonprofits like Small World Initiative or the International Network for Governmental Science Advice. Just I will take a moment to also feature the Small World Initiative that's actually looking to partner with school students to nurture the interest in STEM or interest in looking at soil as a resource for discovery of novel strains that can provide biosynthetic genes for antibiotic discovery, right? So that's something that we're looking to work toward and also willing to partner with countries around the world. And feel free to reach out to me if you're interested in that initiative. Also want to just advertise our institution, the Graduate Center of City University of New York, and current opportunities for students who are interested in PhD programs and several avenues for receiving funding. Please go ahead and look for opportunities for PhD as well as postdoc. Just want to thank everyone for your time. And once again, thank you for the invite. And let us together work together, rise up and help India fight COVID-19 or do surveillance for that and beat antibiotic resistance. You can reach me at these Twitter handles, email, and I also have a Facebook group. And this is the link to Chris Mason's lab as well. Thank you very much. I'm happy to take questions.
So thank you. Bye. It was really extraordinary talk. I was amazed to see a lot of work you have been done, like so far, like a decade of work, right? From Rob Knight to Chris Mason lab. That was amazing work, information.
Thank you.
I hope students were enjoyed the talk, entire session, I guess. It was nicely narrated a story, how from the earth as well as how we are looking at the microbiome, both earth, human, as well as like combination of the urbanization. Wow, that's a wonderful story. Thank you for the informative talk. Let me take this question. There is no question so far yet.
I see some text notes on the chat.
I do see, sir, but
Not.
so
Yeah.
let me take some question like in different aspect like so if a student is doing some microbiology, if he or she wants to do some microbiome study, what kind of skill he or she should acquire during his master or during his PhD?
Sure. Yes, wonderful. Yeah, it's a very, very critical, important question, actually. Good one, Dr. Ganesh. So I think there are two different to doing microbiome. Like I said, a lot of the microbiome efforts that have been done are interdisciplinary. They are teamwork-based projects. So based on your inherent aptitude, you can choose to be a part of that specific module that you want to be involved with. But however, having said that, there are key parts to that. One is, as a student, you are trained on the bench a lot more. You are exposed to laboratory protocols. You're exposed to the art of asking questions based on experimentation. At the same time, several of these inferences would not have happened but for the bioinformatic approaches or the bioinformatic tools that have been developed. Using biologists, several of these computational tools have been developed by researchers who have very uniquely built these tools or algorithms that can help process or understand the data that you are generating from these steps. So the understanding of study design becomes very critical, or experimental design becomes very critical, because that will actually inform the nature of data that you would generate, the data that you generate from the hypothesis that you're framing. To train, you can choose based on your aptitude. Like I said, you want to go on a lab-only basis, but also at the same time, keep your lens wide open to understanding the data that you are generating from your experiment, and work alongside people who are computationally very adaptive, which means they can take that front where you're only involving yourself with handling the data output that comes out of such experimentation. So, or you can work together as a team. So you can, if you're more focused on programming, you like doing software development, you like logical progression, you can actually go into those areas where you can build new open source tools or new add-on tools to the existing set of tools that can help further resolve data sets. For instance, Dr. Vinod Skaria had a very excellent presentation on the assembly of COVID genomes. And one key thing that he did was, I really want to commend his group on that, is making it more inclusive, more available to people from such as students who had the aptitude and the interest to work on the phylogenomics of viral strain divergence. The same way, you can actually look for these open source efforts where you can contribute with your PI, with your research lab heads, to certain efforts on that front. For instance, even this effort that I'm talking about for Metasub India, we can have a way where Shastra students or can contribute to that, right? So you can have students who are taking on different modules. One student group is working on sample collection. On that, one student group is involved with the sequencing process of something like DNA extraction protocols. And then another group is actually focusing on metadata collection, which is actually one of the most critical element for doing microbiome work. As initially, because the environmental information is actually critical to understand the patterns of establishing linkages between samples across cities or within a single city itself. And that actually will be very critical when you're trying to provide a biosurveillance input to the authority. So there are different parts to it, but more than ever now, the understanding that each and every part is critical and important has to be there. Because if you're only doing working in the lab and not focusing on like understanding the computational data, then you are at a disadvantage. What I'm saying is, even the person doing only programming has to communicate with the person who's in the lab or who is doing the protocol. So there needs to be an end-to-end understanding. But if you're very focused on one, if your strength is one area, stick to contributing in that at the maximal level, but also derive knowledge from the other level where you're able to complement. If there is a challenge or if there's something that is required, you're able to pitch it. That's the way the teamwork will happen, and several exciting things can occur. Yeah.
Thank you, Bharath. One question from Ramakrishnan. Yes, does increased demand on livestock industry a reason for spillover of these microbiomes?
Yes, that's of these microbiome. Okay, is he referring to the CoV2 or AMR? Maybe he's referring to AMR, because demand on the livestock industry is one of the reasons for increase in AMR exposure from the environments. More specifically, if you're looking at dairy consumption or even like the cholecystine introduction to cattle or even to chicken meat, right? So there is a trickle down effect from that because people are also exposed to that in the environment. Endocrine disrupting processes are happening because of exposures to such antibiotics. And increased in the Western countries, for sure, there is a tremendous demand on beef consumption and livestock where they are overusing hormonal supplements, they're overusing antibiotics in those cases. That is actually having a problem. And in developing nations, I think the problem is a little different. It may not be livestock alone, but there is also this demand for instance, I think dairy is one big challenge where they just want to mass produce dairy from these livestock, and then they keep certain livestock contained, and there are certain treatments that they are practicing which are not conducive to the growth of the livestock. So those are some of these challenges that are present, which need to be addressed.
Okay, thank you, Bharath. Thank you. Is there no questions? So then we will wind up this session. I thank you again, Bharath, for your extraordinary presentation.
Thank you.
I hope this is interesting for all the students and make them some, interest among on the microbiome topic. So
Thank you, and look forward to collaborating with Shastra. And I wish all the students all the best and stay safe. And please wear your masks.